Hi everybody, my name is Anjali Serpa. I am a third semester student of medicine, and today I'm going to explain you an article about Burkholderia cepacia complex outbreaks that have place in the United States during the years 2016 and 2017. First, we have to know a little about Burkholderia cepacia complex. It is a gram-negative fruit-shaped bacteria that we could find in soil and water. It's an important human pathogen that causes pneumonia in patients with lung disease like cystic fibrosis or in patients that are immunocompromised. This bacteria could be spread by two types of via. First, the indirect via, that it could be caused by environmental surfaces, and the second via, that is the direct, could be patient to patient but is less common, or the via that happen in the cases that is by contaminated sources of food. The objective of this article was identify if the Burkholderia cepacia complex was the cause of this infection and also identify additional cases that could help to the investigation. Materials and methods. First, we have the blood culture, that is a technique that we use to check if in our blood we have different or strange microorganisms like bacteria. It consists in first take a blood sample and then in the laboratory we add each blood sample to a broth. It helps the microorganisms or the bacteria to grow up. If the blood culture is positive, it means that you have a bacteria or something strange in your blood. But if this is negative, it means that you are healthy. The second method that used in this investigation is a pulsed field gel electrophoresis. That is a method that allows the scientists to make like a fingerprint of the DNA of a bacteria to compare if it is present in other samples. To make a PFGE, first we have a blood sample and with it we made a blood culture. Then this blood culture is added to agarose and then we broke or released the cells so we will got the DNA free in this agarose. Then filed separate this DNA according to the size. And the last step is that we observe this under an ultraviolet light and a camera take a photography and storage it in a computer. And the last method used in this article was the gram stain. It is a method that helps us to identify or to distinguish two large groups of bacteria, the gram-positive and the gram-negative. If the bacteria take a coloration like violet, it means there is a gram-positive bacteria. And if the bacteria take a pink or less coloration, it means there is a gram-negative bacteria. It helps us to the treatment of these groups of bacteria because it have different answers to different drugs. Results. First, we have the pulsed field gel electrophoresis, where we could see 11 patterns of isolates from patients that were infected with the bacteria compared with one isolate from a saline solution pre-charged that was unopened. They identified the Burkholderia cepacia complex from a pre-charged solution with a culture and gram stain, and then they compare these results to the samples of the patients that were hospitalized. So they conclude that the bacteria was identical. They quantified the cases in a bar diagram and of the total of cases, 59, that corresponds to the 36%, occur before the notification of the public health authorities. Cases were identified in 5 different states, and the median age of the positive culture was 72 years old. The 81% of the patients were hospitalized, and also there were 16 late ones cases that had the first positive blood culture. And of these 17 patients, there were 10 that were typed by PFGE and all matched with the outbreak of Burkholderia cepacia. Discussions. Now I am going to present 3 affirmations of 3 different authors, and we could see that all the affirmations um, are shown in the article. Conclusions. And finally, we could say that these articles and investigations give us information about Burkholderia cepacia complex and the outbreaks that had happened. So we could have more information to prevent it again. And also we could say that pulsed field gel electrophoresis and blood culture help us to identify if the samples correspond to Burkholderia cepacia complex.